Cleft palate syndrome is a result of a malfunction in embryonic development, specifically during palatogenesis, which occurs during the 4th to 12th weeks of pregnancy, which fuses the face at the midline.
Neural crust theory explains the neural crust cells turn into several types of cells, including connective and neural tissues in the skull and face. These cells migrate from the sides of the face towards the midline to maintain an epithelial bridge across the midline, which aids in fusion. Mutation can lead to a lack in mesodermal delivery and proliferation. In the maxillary and nasal processes, which cause either an absence of cells or deficient tissue at the midline, which causes improper facial fusion. Cleft palate syndrome are known among the most common birth defects and is present in about 1 in 1,700 births in the United States. The mutation has lower incidence in African American populations with a 1 in 2,300 births incidence, but higher in Native American and Japanese populations. Native Americans had an incidence of 1 in 382 births, and Japanese had an incidence of 1 in 578 births. The variance of incidence across ethnicity is likely due to the passing on of different mutations that prohibit proper embryonic facial fusion, which we'll talk about later. The consequences of the condition are difficulties feeding, speaking, dental complications, as well as high rates of ear infection, alongside the psychosocial effects of having a facial deformity. There are currently no known drug treatments to ensure proper facial fusion in the embryo. The only current treatment is surgical. There are two types of palates: the primary palate, which is in front of your teeth and below your nose and consists of your top lip, and the secondary palate, which consists of the roof of your mouth. In a primary cleft palate, a lack of mesodermal delivery and insufficient tissue for fusion will cause a cleft anterior to the front teeth, resulting in what most people are familiar with being a cleft lip. However, in the fusion of the secondary palate during the 6th week of pregnancy, the palate shelves begin to grow from each side of the face vertically initially, and then horizontally until they fuse in the middle. This is what causes the roof of your mouth to form. Fusion of these two plates begins in the 9th week of pregnancy, and then the tissues harden over the next 3 weeks, until the palate is completely formed for the 12th week of pregnancy. In both of these cases, deficient mesodermal delivery tissue prevents the embryonic tissue which grows towards the midline from each side of the face from fusing. This is due to several factors. It is proposed that the causes are likely a combination of both genetic and environmental factors. Environmental factors during pregnancy, such as exposure to infection, certain medication, alcohol and drug use, cigarette smoking, certain vitamin deficiencies, can all lead to increased incidence because these factors either inhibit the pathways needed for proper embryonic development or cause a mutation to the genes required to recruit necessary proteins to properly facilitate the development. Mutations in the TBX22, PVRL1, IRF6, and TGFB3 gene have been identified with increased incidence of cleft palates. However, today we will just discuss the mechanism for how mutated TGF-beta3 gene causes cleft palate syndrome.
There are several genetic and environmental factors that are related to palatogenesis, but in this video we will be focusing mainly on the gene TGF-beta3, which codes for the transforming growth factor beta-3 protein and is expressed in the medial edge epithelial cells, which are cells involved in palatogenesis that eventually die to allow palatal shelves to come together. In wild-type cells at the molecular level, the TGF-beta3 protein is a ligand that has a receptor in the plasma membrane. Once the ligand binds to the receptor, another ligand called BMP4 binds with its receptor, two signaling pathways can be activated, both of which result in the transcription of genes necessary for palatogenesis and correct fusion of palatal shelves. In one pathway, this signal is communicated between 6 SMAD proteins. The TGF-beta3 receptor phosphorylates SMAD2 and SMAD3, and the BMP receptor phosphorylates SMADs 1, 5, and 8. All of these SMADs are called R-SMADs, and once activated, they can gather in the nucleus. SMAD4 needs these 4 SMADs to bring it into the nucleus with them, so they associate and travel together. Once in the nucleus, the SMADs form complexes and bind to regulatory regions on DNA and can act as transcription factors, regulating the expression of certain genes. Another pathway is also activated, which indirectly has the same result. The TGF-beta3 receptor and the BMP receptor can phosphorylate the TAC1 kinase, which then phosphorylates JNK and P38 kinases, which phosphorylate and activate the transcription factors AP1 and ATF2. The BMP receptor can also result in ATP being converted to cyclic AMP, which then activates protein kinase A. And protein kinase A can phosphorylate the transcription factor CREB. The activation of these transcription factors results in the expression of genes that are important for palatogenesis. For example, CTGF increases the proliferation of palatal connective tissue, and IRF6 causes the edge epithelial cells to displace and die, which helps two palatal shelves come together. FGF4 and FGF6 help with tongue differentiation and formation. As a result of these signaling pathways, palatal shelves are able to fuse and the tongue is able to drop so that the roof of the mouth has no gap or palate and the tongue forms correctly. Individuals who are homozygous for impaired TGF-beta3 functioning have cleft palates. Impaired TGF-beta3 proteins come from mutations in the TGF-beta3 gene. These mutations can arise spontaneously, but they are more likely to occur due to DNA damage from alcohol, smoking cigarettes, or nutritional issues. If the DNA is not repaired error-free, the gene can acquire a mutation, and if this mutation does not result in the same protein being made, then there will be no ligand to correctly bind with the receptors. As a result of this, the receptors cannot phosphorylate any SMAD proteins or kinases, so no transcription factors will be activated and no SMADs will bind to regulatory regions. Due to this, the proteins necessary for the fusion of palatal shelves will not be made, and the person will have a cleft palate and likely other facial deformities.
So far, the only treatment or therapy options available for individuals with cleft palates are surgery, speech therapy, or alternative feeding strategies. In order to prevent or minimize the harm done to people from mutations in the TGF-beta3 gene, we propose a drug that would provide the body with a molecule that has a similar conformation to the TGF-beta3 protein. This could potentially help the patient because with this drug there could be something to bind to the receptor in the absence of a functional TGF-beta3 ligand, so that the signaling cascade would still occur. Because structure denotes function, if researchers could create a molecule close in shape to the TGF-beta3 and deliver it to people's cells, then it would be able to bind to the receptor and induce the same response. The SMAD proteins would get phosphorylated and therefore would be able to accumulate in the nucleus with SMAD4 and increase expression of important genes. Additionally, TAC1 could get phosphorylated and then phosphorylate JNK and P38. These kinases could then phosphorylate and activate transcription factors. Finally, it would be possible for ATP to be converted to cyclic AMP so that pKa can be activated, so that another transcription factor can be activated. The result of this drug would be the transcription of the genes necessary for palatogenesis and successful fusion of the palatal shells.